Hi, my name is Donghan Yang. It is my pleasure to give a brief overview of our recent paper in MRM titled Intracellular Water Preexchange Lifetime in Neurons and Astrocytes. The main subject of this study is the transatomal water exchange. The water transit across the cell membrane between the two major water compartments in the living system, namely the intracellular and extracellular compartments. This always ongoing water exchange is of fundamental importance in cell physiology. And more relevant to the magnetic resonance community, there exist many MR methods that target the biophysical properties of either or both of the intracellular and extracellular water. For example, some diffusion-sensitive MR methods are designed to measure the compartment-specific diffusion properties, say apparent diffusion coefficient, and to track their changes during injury. And how fast this transatomal water exchange is compared to the characteristic time of the MR measurements will determine the meaning and accuracy of the ADC results. Another example can be found in dynamic contrast enhanced methods. In some advanced DCE models, the kinetics of water exchange across cell membrane can be either one of the target parameters in the measurements or an important prior information in the modeling. And again, how fast this water exchange is will determine the detailed conditioning of the experiments or modeling. We have listed some very good reviews and research papers on these topics. Please go to our full-length paper for the details. The target of our paper, the intracellular water pre-exchange lifetime, is a very commonly used parameter to describe the timescale of the transatomal water exchange. In a widely used two-side exchange model, the water transit along either direction across the cell membrane is assumed to be a first-order exchange process, and the associated exchange rate constants are denoted here KIE for the intra to extracellular direction and KEI for the opposite direction. When this two-side exchange system is at equilibrium, as in the living system, there is no net change to the water volume fraction in each compartment, denoted by Fi and Fe here. And the intracellular water pre-exchange lifetime is defined as the inverse of KIE, and it is the characteristic residence time of water molecules in the intracellular compartment. We have briefly shown that the timescale of transatomal water exchange, represented by tau I here, is an important parameter in many MR methods and models. And many of these methods target the central nervous system. But there are very few direct measurements of tau I in the central nervous system. And this is the goal of the study in our paper. To be more specific, we aimed to separately measure the tau I for different cell types in the CNS. To do this, we have developed an in vitro system. We nicknamed it Brains on Beads, where the two major types of CNS cells, the neurons and the astrocytes, which is the most abundant glial cells, can be separately cultured on the surface of plastic microspheres. And now we're looking at the immunofluorescence images of this Brains on Beads system. And this is a case of a mixed culture of these two types of cells, where neurons were stained in green and astrocytes in red. We then transferred these cell-attached macrobeads into a perfusion system compatible with our 12T MRI scanner. The layout of the central unit of this perfusion system is shown on the upper left, and the MR measurement we took was a slice-selective inversion recovery spin echo spectroscopy, and the MR excited slices were put perpendicular to the global flow direction, and the scanning direction was from bottom to top, which was against the flow direction. For the inversion recovery in this perfused cultured cell system, the apparent R1 of the extracellular water will be a large value due to the time of flight effect through a very thin slice, and this large flow-dominated apparent R1 for the extracellular water will bring the whole system into the so-called slow exchange limit. where the inversion recovery curve will have a bi-exponential form. And by bi-exponential modeling of the curve, we can derive the effective R1 for the intracellular water, which consists of two terms: the intrinsic R1 for intracellular water and KIE, the exchange rate constant along the intra to extracellular direction. The first term, R1I, the intrinsic R1 for intracellular water, can be approached by a separate inversion recovery measurement in cell pellets, and we have described the details of this measurement in our paper. And in this way, we can derive the KIE, which is essentially 1 over tau I. Let's keep this picture in mind and look at the actual inversion recovery data we collected from this perfused cell culture system. And here's figure 4 from our paper. The inversion recovery curve was well modeled by the bi-exponential model, but not the mono-exponential model, as expected for a system in the slow exchange limit. And we could derive two relaxing components from the bi-exponential modeling. The rapidly relaxing component was assigned to the large apparent R1 for the extracellular water, and the slowly relaxing component was assigned to the exchange modulated effective R1 for the intracellular water. Please refer to our full-length paper for the details of the data modeling and assignments of relaxing components. Now let's quickly jump to the key results of this paper. So figure 5 shows the tau I measured separately for neurons and astrocytes. It's about 0.75 second for neurons and 0.57 second for astrocytes. So this means on average the water volume is shuffled once to twice per second between the intra and extracellular compartments for these cells. And there are two take-home messages from these results. The first is the intracellular water pre-exchange lifetime for neurons and astrocytes are different, and this needs to be taken into account in the sophisticated biophysical models for the CNS. And second, the tau I for both neurons and astrocytes are relatively large in the whole range of tau I's measured for different cell types, especially compared to red blood cells, which has a tau I on the order of 10 milliseconds. And this relatively large tau I essentially, you can say, gives more room for many MR measurements to be engineered into the slow exchange limit or slow exchange regime. And of course, when using these values for modeling and in vivo measurements, one needs to take into account of the change in the intercompartmental water fraction ratio in the in vivo system when compared to our in vitro system. And we have discussed this point in our paper. And in addition to that, the tau I for astrocytes here is an important finding, because in some simplified CNS models, the astrocytes or glia in general are sometimes considered part of the extracellular water, where the transcytomal water exchange in glia is assumed to be fast. But our results show that the time scale of transcytomal water exchange for astrocytes is on the same order as that for the neurons. Therefore, the astrocytes' intracellular water should be considered separately from the extracellular water in the brain. We have looked at the scenario where tau I was measured for cells under normal, healthy conditions. In another scenario, we cut off the oxygen and glucose supply in the perfusion system. And this oxygen-glucose deprivation condition is a widely used model in culture cells mimicking the ischemic injury in the brain. Our study showed that the tau I for both neurons and astrocytes decreased significantly within 30 minutes of the onset of OGD condition. Although this study was not designed to identify the exact biophysical mechanisms behind this decrease in tau I upon oxygen-glucose deprivation, we have discussed some possible factors in our paper. The observed decrease in tau I here will have an impact on the previously mentioned applications to the MR measurements in the CNS to the extent that the oxygen-glucose deprivation can capture the essential features for ischemic injury in vivo. To draw the conclusion, in our study, the intracellular water pre-exchange lifetime was separately measured for neurons and astrocytes, and we observed that the tau I decreased significantly under oxygen-glucose deprivation conditions. The results presented in this study will provide useful prior information for designing MR measurements that are based on the slow exchange limit two-side exchange model. For example, the compartment-specific ADC measurements in the brain. We'd like to thank the fundings that supported this study and the general support from the home base of this study, the Biomedical Magnetic Resonance Laboratory at Washington University. Thank you for your attention, and we look forward to more discussions on this topic.